Okay. Well, looks like we have quite a few folks logged in this morning or this afternoon or evening, depending on where you are in the world. Welcome to the ClinGen Ancestry and Diversity Forum, if this is your first time attending. And we are delighted today to have Anna Lewis, who's a research associate at Harvard's Safra Center for Ethics, where she works on the ethical, legal, and social implications of genomics. She's also a member of the Genomes to People research program at Brigham and Women's Hospital in Boston. And she's originally trained as a computational biologist and was previously employed in the genetics industry. And her LC work is deeply rooted in scientific and technical developments. So we're delighted to have you here today, Anna, talking to us about your recent paper with some folks, probably some co-authors who are also on the call today, getting genetic ancestry right for science and society. So welcome and take it away.
Thank you so much, Alice. What a great delight to be here. I'm a great fan of everything ClinGen does and your group in particular, of course. So this is a great pleasure to be here. So as Alice mentioned, she asked me to speak to you off the back of this preprint that we have, Getting Genetic Ancestry Right for Science and Society. And so this is the work of this group that we have based at the Safra Center at Harvard and under the intellectual leadership of Danielle Allen, who's a political philosopher currently running for Massachusetts governor. So she wears lots of hats. And Benjamin Neal, who's a statistical geneticist based at the Broad. And the work I'm going to be talking about is also in conjunction with a team of research associates that I work with here, and with an interdisciplinary working group that we've assembled to give us input onto the direction of the work. And key to our approach is this recognition that genetics is a science at the intersection of several disciplines. So anthropology, sociology, demography, just to name a few. And this is a story I'm going to tell you in 3 parts, and the opportunity, the danger, and the path forward. And we touch a little bit on the path forward in the preprint, but I want to talk some more about how we see that path forward, drawing on some descriptive work that we've been doing. And this is preliminary stuff, so you guys are actually going to be the first ones to see it. So I'm particularly interested in any feedback or questions you might have. And the punchline of all of this is we see there being a scientific and ethical imperative to embrace a multidimensional continuous view of ancestry and move away from continental ancestry categories. Okay, so we'll start with the opportunity. First up, ancestry of central importance is a debate about the use of race as a variable in medicine. So we've seen just recently quite a lot of focus on the ways race is inappropriately baked into health algorithms. This is a very live debate. This paper from last year, reconsidering the use of race correction in clinical algorithms, one of the things they said is the appraisal of whether the use of race or ethnicity is appropriate in algorithms that include race correction will require further research into the complex interactions among ancestry, race, racism, socioeconomic status, and environment. And here are 2 pieces sort of in response to that one of which, titled Race and Genetic Ancestry in Medicine, A Time for Reckoning with Racism, argues for the continuing use of both race, ethnicity, and ancestry, and saying race ethnicity categories used in biomedical research and clinical practice are broad and less precise than ancestry. Ancestry is a fixed characteristic of the genome. So I'll have a bit more to say about that in a second. And also this piece, Embracing Genetic Diversity to Improve Black Health. The ultimate goal, we believe, would be to replace race with genetic ancestry in an evidence-based manner. And we've also seen this with the American Medical Association turning towards genetic ancestry as we reconsider the way race is used across biomedicine. And also we see ancestry appeal to for this is in in terms of response to viral infection. This is from last week in Science, where they're pointing to results being consistent with a potential contribution of genetic ancestry to observe COVID-19 health disparities. So that's sort of outside of genetics. That was the increasing relevance of ancestry as a concept. But as we all know, it's also of increasing relevance to applied genetics in the context of rare variant interpretation, where population allele frequency data is all important to helping establish whether or not a variant is linked to a particular phenotype. This original 2015 publication, which still dictates the way that things are done in laboratories, states that populations should be race matched, whatever that might mean. But of course, it's with polygenic risk scores that this has really come to the fore. So if we look at the distribution of unadjusted polygenic risk scores, and for those of you not super familiar with polygenic risk scores, these sum up the small effects of many variants associations to a phenotype of interest. So you get out a number that you can then process in various different ways, maybe of potential relevance to the clinic. But if you just plot those numbers before you do anything else to them, you see that you have these different distributions by population group, and it doesn't actually matter how you carve up those population groups here based on 1,000 genomes, defined populations, here based on self-identified race within a biobank. You see these differences in distributions. So this is potentially problematic. And moreover, if you then go and see how well those scores predict the phenotype of interest, you see that we have differential performance of these scores by these population groups. Also speaking to the increasing relevance of ancestry within genetics, here's a paper out again last week in the American Journal of Human Genetics, where the authors trace the use of all these terms within the AGHG publications over the last few decades. And you can see ancestry rising in importance, as well as some of these continental categories rising in importance. And I think it's also important to note that ancestry is the predominant framing for calls for diversity and research participation. I feel like almost every genetics talk I go to ends with a call for the need for diverse participation in genetics research. There's an incredibly long history of calls similar to this, although there seems to be a real sort of contemporary push. And these contemporary calls call for diversity variously refer, if they refer to anything at all, to diverse individuals, diverse populations, or diverse ancestry or ancestries. But while some papers mention the relevance of multiple types of diversity, in practice, diversity most often means ancestrally diverse. And I'm with one of our research assistants, Nanika Ghosh, we're thinking through this sort of diversity imperative in genetics research. So here's the opportunity then. It's to reconceptualize human biological difference. So no small thing. We've got this moment when people are reconsidering race as a biological variable within clinical medicine and within applied genetics. We've got the sort of relevance of this concept rising to the fore. So if that's the opportunity, what's the danger? So we tend to refer to race, ethnicity, and ancestry as these three different sort of sets of population labels. But the thing is, we don't live in a world where these are nice and neatly separated. Instead, they're all kind of merged together. So let's have some examples of that. As I'm sure most of this group knows, it was previously proposed that the census categories, which dictate so much of the way that we categorize people in this country, it was an update to that was proposed under the Obama administration that would combine race and ethnicity and ask people to select any combination of these that apply to them. That didn't go through under the Trump administration, and probably somebody on this call knows better than me what the current status of this debate is. So that was race and ethnicity being merged together. Ancestry and ethnicity are often not distinguished. Here is something from My Heritage DNA, where they're really talking about the results that they give in terms of ethnicity. Here's another figure from a Martin et al. Paper. I had another figure from it earlier, and this was the paper that framed all things in terms of these population groups, these ancestral population groups. And then this is how it got picked up in the media, referring to racial breakdown of these groups. So we get this slide between the sort of population categories that geneticists used and race. And I've also observed this this complete slippage in sort of genomic implementation science work that I do. And then over the last few decades, we've seen a rise of continental labels. This is from a wonderful paper by Aaron Panofsky and Catherine Bliss, where they chart the use of population labels in this case, Nature Genetics. And you see that the main thing that's changing here is the use of continental labels to describe studied populations. In genetics practice, we also see a big sort of merging of certain concepts. There's a big old project called eMERGE4, which is a genomic implementation science project, where something that we're doing is using these 4 categories: African, Asian, European, Hispanic, but those population labels, they can come from anything. They can come from self-reporting EHR extraction and or analysis of genetic ancestry. So it's all over the place. And yeah, we see this when you actually go and talk to physicians and patients who might be interacting with some of polygenic risk scores, for example. This slippage between race-based language and continental ancestry language is everywhere. It's just endemic. So this is the danger then, that continental ancestry categories represent the same old typological thinking. So one of the problems with racial categorization was this idea that humans come in these distinct type of essences. There's a small number of those groupings that we can sort people into, and adopting continental ancestry categories is the same sort of typological thinking all over again, just dressed up slightly differently. And I want to emphasize that admixture, embracing a notion of admixture, is not the solution to this problem. It's part of the same problem, because there you're still thinking in terms of these discrete groups, these sort of pure types, and you're just allowing some individuals to be mixtures of those otherwise pure types. And typological thinking has all sorts of problems from an ethical point of view. History teaches us that very well. But we also think that these continental ancestry categories hold us back scientifically. So for one, sort of if you look at population structure of individuals in sort of some real world populations, you don't get just discrete clusters. Instead, you see that people are spread across this continuous space. So as many others have noted before, a lot of the sort of ways in which certain groups look like islands in genetic structure space follow from the way in which sampling was done to get those data. And so these colorful islands here are from some of the big population databases. And all the gray dots are from a biobank, in this case from New York City. And you can see that individuals sort of populate a lot of this space. So ancestry is continuous, not categorical. And ancestry is also a historical concept, not one that's about essences. This is an unsatisfactory picture in many ways, but the point is that we all have ancestors from every point in our past, and just taking one slice through this complicated, not tree structure, but something that's sort of like a braided bush or a braided stream as an analogy that some people have used. If we just take, if the sort of continental ancestry slice is at most a gross oversimplification of one time slice of interest. So ancestry is a historical concept. And I just wanted to quote this directly from our preprint, because this is for all of those of you who have written things in combination with a lot of people. There was this is very carefully worded and adjusted with input from people from all sorts of different disciplines. So let me just go for this. So an individual researcher's use of continental ancestry categories is not in and of itself racist, but the cumulative impact of this practice has led to and sustains racism. Typological thinking about human difference has had damaging social consequences. Continued reliance on continental ancestry categories contributes to failures of inference, miscommunication between fields, and reported findings that are rooted in reductive and limited ways of understanding human difference. These are likely to exacerbate medical stereotypes about individuals and groups, contribute to health disparities rather than addressing them, and reify biological misunderstandings of race as biological. And so this is the sort of reason why we have this scientific and ethical imperative to move away from continental ancestry categories, which takes us to the path forward. And in this preprint, we sort of gesture at some of the component pieces, and I want to start there and then think about where we go from here. But maybe I'll pause here and ask if there's any questions to this point. Alice, if that's appropriate, or if you prefer that I just plow on, just say.
It's totally up to you. If folks have questions and you want to stop now, that's totally fine. I think in the forum, people tend to go through to the end and then have discussion then. But if you want to pause.
Yeah, well, anybody want to jump in? Because if not, I will just plow forward. Okay, I'm plowing forward. Okay, so what is the path forward? The solution will require addressing the issues with how ancestry is conceptualized and used across the entire biomedical research ecosystem, not just within each subfield of biological science. That's an appeal back to this interdisciplinary field that is genetics. This will involve the development and operationalization, what a word, and hence widespread use of a more complex notion of ancestry, one which disambiguates what is meant by ancestry, that wherever possible does not treat it as a categorical variable and treats ancestry as reflecting a historical process. So I'm gonna take the second of those three first, avoiding categories. So I made the case to you earlier why these categories were problematic, both scientifically and ethically. So we believe that methodologies that avoid categories are preferable to those that employ them. And I think increasingly we are seeing methodologies that avoid putting people into categories. So here's a classic one from Jen Roderick et al., where they're controlling for principal components even across continental ancestry groups in their genome-wide association study. And the motivation is exactly as we've been talking about, given that their participants reside on a continuum of genetic ancestry, rather than discrete population groups, a joint analysis was optimally powered and the most parsimonious way to allow for heterogeneous variance across populations. So this is an alternative to where you just split everybody in your data set up into groups based on their genetically inferred ancestry or some notion of self-reported in combination with some sort of methodology for excluding outliers, showing that you could get just as good, if not better, results if you if rather than stratifying your analysis and putting people into buckets, you control in a continuous fashion behind the scenes for the types of differences that you might be worried would confound your results. Here's another example. I showed you earlier distributions of polygenic risk scores where the distributions look different by these population groups. Here's another example. In this case, this is for chronic kidney disease. So you see the unadjusted scores look all over the place. And that won't do for clinical applications because we don't believe that, for example, the differences in mean between these populations are biologically meaningful. And if you have some kind of a rubric, such as report high genetic risk to those in the top right-hand tail of these distributions, as is being done in this big emerge study, for example, then you're going to predominantly just select people who are at high risk. In this case, it would be the Europeans, but it could be any one of these groups. So this is not a good state of affairs. But it turns out that you can control for the differences that are driving why these curves look different in a continuous manner behind the scenes using principal components in this case, and get some distributions which are much more closely aligned. So that is what's beautiful about this methodology, is you haven't had to be, okay, let me find out what the mean and the standard deviation is for each of these separately and then align them. No, you've just employed the same methodology to all of them. And there are some other examples in an increasing number coming online. Okay, so this other thing that we wanted to appeal to, that ancestry should reflect historical process. Here are a couple of examples of a more historical use of ancestry. So rather than always appeal to continental ancestry categories, this lovely paper links to our Neanderthal heritage. So the main GWAS hit for COVID severity, somebody noticed that it was on a haplotype whose length was characteristic of introgression from Neanderthals. And then they went away and looked at it, and lo and behold, they at least make the case that that this is a sort of Neanderthal variant, which some of us have inherited. So their focus was not on this sort of time slice where continental ancestry categories might be the most obvious one. It was something further back in time. And to give another example, this is a preprint, I haven't seen it published anywhere, where they modeled all Europeans as a combination of 3 source populations. And then went looking for correlations with COVID severity. So it's just to give another example of how thinking about ancestry in a more historical way can suggest other research questions and things to go look for. And I think we're starting to see more time slices coming from the consumer genetics world. So this kind of picture where one's genetic ancestry is compartmentalized into these continental ancestry buckets is probably familiar. As is this other time slice of how much Neanderthal DNA you have. But this is a newer thing from 23andMe, where they're using the genotype data to suggest more recent history. This time they've done this for many of the places in the Caribbean and Central America. So this is this idea of, there are multiple time slices of potential interest when it comes to ancestry. And then the third thing that we had said in the preprint was the importance of disambiguating ancestry. And this is where I want to start drawing on some empirical work. So why is it important to disambiguate ancestry? Well, without clearer concepts, we're going to get inaccurate generalizations, unclear comparisons, and also generalization and comparison claims getting lost in translation across disciplinary boundaries. And I just want to acknowledge before we dive into this, that there's ancestry and then there's genetic ancestry. And some people think that these are very clearly delineated. So for example, at a GA4GH meeting that Alice, for example, was speaking at, Charles Ratimi said, we have to distinguish between colloquial use of the term ancestry. Ancestry is genetic. We don't know the sources of our ancestry. But we do have these two different concepts in play, ancestry and genetic ancestry. And here I just want to double down on the fact that genetics is a discipline at the intersection of several disciplines. So the descriptive work, the empirical work that we've been doing is really rooted in this idea. And I want to share some preliminary results. These were qualitative interviews that I did along with Santiago Molina, who's now at Northwestern University. And I'll tell you how we decided who to interview in a minute, but we interviewed a bunch of diverse researchers, and one of the things we asked all of them was, what does ancestry mean to you? And these were all researchers who actively use the concept of ancestry in their work as a sort of central piece of their work. So, I mean, maybe it would be fun for you each to think how you would answer this question. But I can tell you some of the punchlines. So many people struggled to answer this question. We didn't get the same answer twice, although there were some patterns. And several individuals gave us multiple definitions. And I love this quote from one of our interviewees. We try to define the terms we use, but it's kind of like trying to catch smoke. Oops, wrong way. So we've been analyzing the responses to these, and I think we capture quite a lot of what's going on by picking up on two different dimensions of variation. One of what ancestry is a property of, and the other is what criteria are appealed to. So this is sort of a two-dimensional space of answers that we got. So ancestry being a property of DNA, of an individual, of kin or family, or of a population. And then the sort of criteria that are appealed to is what these have in common could be biology, ethnicity or culture, genealogical connections, and geographical origin. And geographical origin was the largest of these buckets. And I wanted to highlight in terms of the relationship to ethnicity or social identity. So when ancestry was defined as being a property of an individual, it was mostly either explicitly that it was the same thing as something to do with ethnicity or cultural identity or social identity, or explicitly not that. And I just picked up basically the first two quotations randomly, and here are some illustrations. So the first one saying ancestry, it's not just the percentage that you can boil it down to for each individual. There's a lot of, cultural and other social aspects that go into it. Versus it's a statistical concept that we use. Sometimes we're not being that specific because there's no kind of clear borders. So it's a way of kind of conceptualizing something to kind of grasp a certain group of people, but it's very far from concepts like race or ethnicity or something like that. It's just a bunch of people more sharing a common ancestor than another group of people. And we went into this expecting to find quite a lot of differences between disciplines about how they spoke about ancestry. But actually, we just saw so many differences within disciplinary groups, as well, of course, as across disciplinary groups. So within any given field, ancestry is being used to mean many different things. And that's true of genetics as much as of other disciplines. So that gets us to the question, what is genetic ancestry? And if anybody hasn't come across this wonderful paper by Ian Matheson, Elwyn Scally, What is Ancestry? I heartily recommend it. And what they end up arguing for is that genetic ancestry just is this thing known as the ARG, the ancestral recombination graph. And this describes the inheritance of each part of the genome of every individual who's ever lived. The nodes represent the genomes of individuals, and edges represent genealogical connections between them through which genetic material has been inherited. And then various different methodologies relate in different ways to the ARG. So, so we are defining genetic ancestry following that paper, and that paper is basically the only one I know that actually goes and lays out a definition. So an individual's genetic ancestry is the subset of paths through their family tree by which they have inherited DNA from specific ancestors. So it's really, really important to notice two things that means genetic ancestry is not. And the first is it doesn't give any context to those ancestors beyond their genealogical relationships. It's silent on their ethnicity, on their location, their geographical location, on the language they speak, or anything else. And second, it doesn't employ any notion of a population. You don't have any notion of any type of categorical schema at all in this definition of genetic ancestry. And one of the things that makes this hard is that there's lots of related concepts that we need to disambiguate genetic ancestry from. So these include genotype, genealogical ancestry, genomic relatedness, population structure, population stratification, genetic background, identity by descent. So part of the work as we see it is to separate out these concepts and clearly show where the light is between them. So the final substantive thing that I want to speak to you about is understanding the use cases for ancestry. Because I think this is just a necessary component if we want to arrive at some normative recommendations, or frankly, just if we want to understand what's going on. So this, again, is very preliminary from a qualitative literature analysis led by Becky Dowder, who is a research assistant on this project and is a postdoc at Penn. And we think, and what we did is we identified just over 200 articles which use the concept of ancestry across lots of different disciplines. And I told you I'd mention who our interviewees were. Well, our interviewees were a subsample of the authors of these papers. So we're combing through these papers to look at lots and lots of different questions, but I just wanted to focus on use cases for ancestry. And I think these fall into 5 different buckets. So ancestry is used as a variable at various points in processing pipelines. It's used in connection with the generalizability of findings. It's used to connect to health outcomes. It's used to understand aspects of how history influences the present. And it's used to compare ancestry to self-identified categories. So I'm just going to take you through these. So ancestry is used as a variable at various points in the processing pipelines. Here are just some examples. To choose the chip that you're going to actually use to genotype your participants. I mentioned this one to exclude outliers. So famously, for example, in the British Biobank, they employ the concept of ancestry to exclude individuals from their core white British sample. It's used to choose a subgroup of interest. It's used to do ancestry-specific QC in a variety of ways. It's used to do ancestry-specific cutoff values for other variables that a study might involve, for example, BMI, where a threshold for obesity might be set differently in Asian ancestry individuals compared to European ancestry individuals. It's used to select an appropriate statistical modeling framework. A very big one, to control for population stratification and to check for uncontrolled population stratification. And something that we really observe is very often in when researchers are describing how they're using ancestry, they just say, as previously described, we do X, Y, Z. Second bucket of use cases in connection with the generalizability of findings. Well, I already showed you this assessing predictive performance of polygenic risk scores. To improve results derived from just one group. So here, this would be researchers saying, well, we looked at this other data set, which had people with different ethnicities, and we can show you that we did better than if we had just used European ancestry individuals. And also to claim a diverse data set. So it's like, hey, I'm gonna put people into these ancestry buckets or otherwise show you what's going on with the ancestry of our sample in order to claim that I have a nicely diverse data set, which is presumably useful for things downstream. Third, and I'd say this is probably the biggest one, is connecting ancestry to health outcomes. And the most straightforward way of doing this is you get out a variant that you've associated to some phenotype, probably a disease outcome, and then you go and look and see what is the frequency of this variant between groups. With COVID, we saw just literally dozens of preprints doing exactly this, and many of them did this thing, which is just so easy to do, which is to connect allele frequency differences by however major databases are defining populations with race-based health disparities. But there's all sorts of other ways of linking ancestry to health outcomes. Directly looking at a health outcome with ancestry, that can be self-identified ancestry, it can be percentage of a certain continental ancestry categories, and very rarely something else. And it doesn't just have to be health outcomes, it can be also sort of intermediate biological phenotypes. For example, gene expression profiles, tumor mutational burden, that type of thing. Some people are directly predicting health outcomes with ancestry as one of the variables. And quite a lot of people are trying to unpick the contributions of genetic ancestry with either self-reported categories like race or ethnicity, or with environmental factors. And there we found a sort of subset of papers that explicitly set out to show that genetic ancestry does not play a role in explaining some of these observed differences in health outcomes. So just an example of this type of work. The associations of genetic ancestry with complex disease are varied, with some data suggesting that greater African ancestry is protective, whereas other data suggest the opposite. So I feel like this is a particularly messy corner of things that are going on, and I don't think there's any clear guidelines for somebody to claim, oh, this shows that there is an association with ancestry, or that there is not an association of ancestry. There's not much attention to effect sizes, to other things that could be going on, etc. And just to stress, very often the starting point motivating a lot of this work is race-based health disparities. Fourth category: to understand aspects of how history influences the present. So this can be straightforward population history, including stuff which involves ancient DNA. Also in the context of linking to disease status. So to try and understand how population history might intersect with disease status, including talking directly in terms of evolutionary selection pressures. And another use case, which I thought was quite interesting, was to learn more about human remains. So like a standard example might be that you've got some unidentified human remains suspected to have been people forcibly shipped from West Africa, for example, and the quest is to learn more about those human remains. Final bucket of use cases is to compare to self-identified categories. And some people do this without a motivation beyond, we can do this and we can tell you about the results. In some cases, it can be as a QC step. So for example, you might have some cell lines labeled with a self-identified race. You might not be confident in those. I'm thinking of one paper in particular. So they're doing this analysis of genetic ancestry to see if those labels are likely to be correct. Some people want to predict self-reported categories, and there's various reasons why they might want to predict self-reported categories using genetic data, including, of course, in forensics. And another one is to try and understand the evolution of self-reported categories. So sort of that also speaks to the intersection of some of these different ways of grouping people. Okay, so we're doing all of this work because we want to identify the pieces that are needed to help researchers move beyond continental ancestry categories. And I want to emphasize that we think that in some cases, using continental ancestry categories will end up being appropriate and necessary. But the danger we see is just that this becomes the one and only predominant way of understanding human biological difference. And we think there are all sorts of problems with that. So we're working on some normative recommendations, pulling together various of the threads that I just shared and some other ones. We're interested in where there might be missing standards and also missing accessible software. I think that's something that we've really seen. Often there is software, but it's only designed for use by a very specialized audience. And we're hoping to feed into the recently announced National Academies Committee on the use of race, ethnicity, and ancestry as population descriptors in genomics research. And more broadly, we're interested in doing stuff that the community is going to find helpful. So I'm particularly interested to hear from any of you on what types of things related to the types of things that I've been speaking to you think might be particularly useful or fruitful. And I'd also just to end like to say, it'd be a great pleasure to talk with any of you after this on these topics. So if this has sparked anything in particular for any of you, I'd love to hear from you afterwards. Cool. So that's all I've got. I'm going to stop sharing my screen. Cool. I don't know if anybody wants to follow up on the comments in the chat. I'm not sure I get all the context of the first one.
Thanks so much, Anna, for the great presentation. Especially loved your question, what does ancestry mean to you? And it's interesting, you've been asking that to investigators. I've been asking that to members of community action boards and
cool.
and research participants, and just getting a sense of, and the breadth of responses we get is very, you know, is very interesting as well. But I would like to follow up on Laura Fairman's comments here, if she'd like to just speak to that. Laura, if you'd like to first.
Hey everyone, it's nice to be here. Thank you for a very interesting and provoking presentation, I guess. My comment was when you were showing how you could take into account the PCs in analysis without having to, talk about geolocation or any other concept. And the paper showed the PCs by category. So in a sense, I think it's very circular that we try to make the analysis more complex, but there's always one step in our pipeline where we end up still using some category, even, for example, when you're trying to oversample people with a particular pattern of variation. So my comment was more, I want to hear from you is, how we think and then how we do things could be very different, and they can be confounded. And yeah.
Did you mean the ones where it was polygenic risk scores and they look different, and then you do something with the PCs?
There were like 5 PC plots, and you could see the beyond continental grouping. You could see the different PCs, and you mentioned that in that paper they were using the PCs and not talking about ancestry, continental ancestry, for example, when they adjusted whatever work. I haven't read the paper, but that was the point. But on the plot, you had African Americans and then the PCs, and like Hispanic, Latinos, and then the PCs.
Yeah, so there's sort of a use case where you're checking what's going on. Yeah. I mean, there you don't need to control for population structure by using PCs and GWAS. You don't need labels on the samples, but it does help you see what's going on. So I think you're right. There's a kind of like visualization piece and seeing what's going on. That might end up being one of the legitimate use cases for these buckets to get a bigger picture. Yeah, but there you don't actually use the labels in analysis.
I know, but we like that I think that's very interesting. It's like you're still thinking in those terms at some point in your analysis.
Yeah, and I think the one which I find hardest is performance of polygenic risk scores. Like, it's very hard to think about how you capture that without using some categories. Yeah. So I agree. It's just, I mean, there's a question of which category should you be using, of course, but I don't think we're going to be able to fully get away from categories.
So I think the problem has been the lack of, we do science and people are not explicit about how they're using the words they use, so that how we, you operationalize a more abstract concept in your research. And I think there's room for different ways of referring to populations, depending on the level of your question and what are the issues you want to address. So
I'll just say, I think transparency is a minimum bar, but it's definitely not all of the story.
Right.
So thanks for some of the comments in the chat as well to kind of fill out the conversation. I'm going to go to Luis, had a question, and then Carlos.
Yeah, thank you, Anna. Great talk. I have a question more about, perception of ancestry, race, and ethnicity and continental origins of people, people of color like me, a Latino, and white people. Because I have this impression that most of these definitions are driven by white scientists, and I don't know if, how these perceptions are being taken into account when people decide to come up with, okay, we are going to refer this as ancestral recombination graph, or we are going to refer to this as, this other concept. So I wonder how dynamic is the conversation about race and ancestry with non-white scientists, and if you have any idea about what the difference in perceptions about these terms are from whites and non-whites.
So specifically amongst scientists, I can say something that we observed is that scientists of color were often particularly sensitive to questions like, well, but hang on, that means that I'm not represented in the data. Like, you've gone and collected my data and then you've excluded me as an outlier. So that kind of thing. And I'd say two perceptions of lay people. It sounds like Alice is doing something similar, but directly asking non-scientists how they view these things. And I also wanted to mention that we've been doing a big old systematic review of recommendations that have been made in this space, and almost all of them come out of the US. So I think we're getting a sort of a biased view on normative side because of that. But in our interviewees, we tried hard to be international. So we've got a lot of perspectives from South and Central America, for example, which has been really interesting.
Carlos?
Thank you, Anna. I really appreciate the talk and the paper. So I just wanted to make one comment, which is that this is, while seemingly new, not really a new problem. This is a conversation that has been going on at least as old as the field, and I'll point us back to the HGDP papers. When they first came out, there was a whole discussion about clines versus clusters, and I put the link here in the paper. And methodologically, what are you really doing when you go from different levels of K in structure, and what does that really mean, and how do you interpret it? And that is, at least from a design point of view, from what might be called anthropologically defined self-identified groups that you're trying to sort of look at different scales. In fact, the whole work of Cavalli, Sforza, and Edwards is all around building population level trees and allele frequencies to sort of track exactly these kinds of questions. So I think you've reframed the debate in a very interesting direction, but I think it's important to sort of put it into that historical context, because there are some learnings there, both good and bad, that probably want to go back to. I think the second comment is that today, the bulk of the data is simply held by healthcare systems, and so when you look at BioMe and BioView and the Broad's own work on Nomad, there's a sort of practical problem that you're trying to solve, which is there's a gush of data coming at us. What are the appropriate ways to model that data in order to make inference in some very discrete settings, or not in a general use case? We're trying to do GWAS. What's the best way to do this? What's the best way to power this? We're then trying to take those GWAS results and translate them into individual level risk. What's the best way to do that? What are appropriate ways to do that? And the labels in some sense don't matter. You can call them PC 1, 2, 3, 4, 8. You can call them you can use different approaches to do this. You can use log linear models, right? That become sort of a model versus, data problem, and does the model accurately reproduce the data? Is there a generative model? And in some sense, there's also a risk here, which is in letting the perfect be the enemy of the good. You get to the UK Biobank problem of, we're just going to use this to filter out the people we don't want to analyze, and you have the whole other problem, which is undersampling of populations, underpowered studies. And if you use all comers and you don't use stratified sampling, then you're going to get into a situation where obviously the data is going to accumulate in the majority population before it accumulates in the minority population, simply due to numbers. And so I would really urge us to be cautious about how this then translates into, altering ways of working for the simple reason that this field itself made, I think, a mistake when we first started to go into SNP discovery, and we had a SNP panel that Debbie Nickerson sequenced, and it was against the rules of the data use to try to look at ancestry of the individuals, precisely because there were lots of concerns after HGDP about how the sampling and this and that. So this is a very, this is a dialogue, that we've been having for a long time, that's been swinging from one way to the other, and the pendulum's now swinging in a different direction. At the same time, you go to ancestry.com, they've got 2,000 groups that are defined by where people were born, and they do an incredibly accurate job of describing segmental segments. So there's the science here, there's an element of the social use of the data, and then there's the creating of infrastructure for NHGRI, and all of those are at different tensions here. And so I think the work is extraordinarily interesting, and I commend you on it. I think about the implementation side of this, and I would caution us to heed your warning. At the same time, we have to have reasonable alternatives, particularly things that could turn into code and turn into actual process. So anyway, more a comment than a question, but I'd love your reaction.
No, thank you for that. So I do think that identifying the implementation pieces are exactly the level we want to be engaging with, because I think sometimes you're going to get where you're like, we just need to use these categories for this use case for this reason. But then there's other pieces where it can be, no, actually, if we think about this carefully at this point, we can avoid continued reliance on these categories. And I'd say, this is definitely, for people who've been in this field a long time, this is it's very frustrating that we're still talking about these same things. I've definitely got some people in our working group who are like that. For me, I think that some things do feel like it's an opportunity to really have this conversation again. So part of it is the opportunity that I outlined in terms of, in general, the questioning of race as a biological variable in medicine. I think that the rise of ancient DNA has changed things. I think the issues of polygenic risk scores on the genomic implementation science, the clinical genomic side of things, has also changed things. So I think a few, and as you mentioned, just the sheer quantity of data and coming online, all of these things intersect to mean that, we at least feel that there's another moment of opportunity. But thank you for your comments. I agree in general.
This is a great discussion. We have 1 minute left and 2 questions. I'd love to be able to just hear them. So maybe, and as Carolina was first, and then maybe Simon, if you chat yours, so we can kind of get them simultaneously, and then Anna, have you kind of reflect and have the final word.
Sure. So I thank you. I wanted to say more of a thank you, but I'll just leave that there. The question that I wanted to ask is sort of driven by these conversations about polygenic risk scores and also this issue of population stratification, is that the development of so many of these tools and these methods is population based, in the sense that the relevant variants are interpreted out of clusters. And from some of the remarks you said earlier about like the methods that we would want to use remove categorical labels or remove categories, but also it seems like it's not really possible to remove categories. So I wanted to ask, would doing these sort of behind the scenes analyses to try to get away from this categorical issue actually solve the problem, or would it just make it less transparent?
I mean, I hope it's going to help with the problem. And I think when you're dealing with one variant at a time, it's always a bit tricky because it's so easy to go straight to these allele frequency databases. But I think some of the pieces we've got to put in place are, if you are going to do that, then what else do you have to do before you make any claim of that's in connection with health disparities? Because right now it's just like the next step of logic is that, but it's a much more complicated picture, as we know. And absence of talking of effect sizes doesn't help. So I think we should probably discourage just straight away going to look in these allele frequency databases when you've got a variant which is a very small effect size anyway. I do think that looking at individual variants allele frequencies is yeah, one of the big pieces of what's going on. With the individual as the unit of analysis. Oh, interesting, because most people think that going to the DNA is the answer. I'd love it if you would follow up with me by email.
Yeah, that's cool. I hadn't seen that paper before in detail. I think it's one of the problems that we have with labeling is that we have a pretty clear concept of an individual ancestry, that I'm me and I've got my grandparents and cousins and all that kind of stuff, and I'm usually wrong in terms of like actual knowledge, but I've got a pretty clear concept of the individual and how that spans out and broadens. But when we talk about ancestry, the unit of analysis, as well as that popular knowledge versus genetic understanding of ancestry, the unit itself also gets very conflated really early on. We're often talking about different things, talking about population-based ancestry, a population, as in something like way off, like, yeah, even like Neanderthal ancestry, ancestry. And then we have our own, like, where am I situated in comparison to all the people that I would say I'm absolutely related to, even though I don't know them. And I think that gets confused really early on. But what was interesting about what you showed is, yeah, that's like a neutral way of showing individual ancestry. It's got none of those terms. We're happy with that. We can deal with that. What we can't deal with is a collective unit, because we don't have names for it other than race and ethnicity. We just, yeah, and PCA, you know, as Carlos said, you know, you might use like PCA 1 to 8 or 20 or whatever, but as yet, that's difficult to do in popular terms. That's why like 23andMe doesn't do that.
Yeah, I mean, some papers just use colors, like we've got the green group and the blue group or something. Yeah, sometimes that works. Yeah.
Eventually, people go, what's the color mean? Oh yeah, it means South American.
Yeah. Yeah, yeah.
And then you're going to get that slippage. You know, as you call it slippage, I mean, slippage is a negative term. I think we need to use like a more, that's what we have available to us at the moment. That's why I think the individual one is interesting, because it doesn't use that type of language. But that's 10:04 and that's me ranting. Okay, but we'll.
No, thank you.
Very interesting.
And thank you, everybody.
Thank you so much, Anna. We'll obviously keep this conversation going every month on the Ancestry and Diversity Forum, so happy to have you here. And anyone else who is interested in making more comments, perhaps in the length of 45 minutes or so, please volunteer to present on a future forum discussion or nominate one of your colleagues. Have a good week.